She was 20 years old. She weighed 16 pounds. She had never spoken a word. She had never walked. She had never grown a single permanent tooth. Her body was the size of a toddler. Her face was the face of an infant. And yet she had lived through 2 decades of birthdays. 2 decades of strokes, 2 decades of medical crises that should have killed her many times over. Her name was Brooke Greenberg. She was born on January 8, 1993, in Baltimore County, Maryland. And from that moment, her body did something no medical textbook could explain. It refused to age. Not the slow, graceful slowing of cellular wear. Brooke's body did not slow down. It simply stopped. As if some master clock inside her had been broken at the factory and never restarted. But that was not the strangest part. Doctors who studied her could not categorize her. Geneticists who sequenced her DNA could not name what was wrong. By the time of her death, she had been examined by some of the most respected aging researchers in the world. None of them had an answer. What they did have was a hypothesis. And it would change the way scientists thought about human aging forever. The pregnancy had not been entirely normal. Brooke was born several weeks premature, weighing just 4 pounds. She had a dislocated hip. She had a problem with her stomach that required surgery in her first weeks of life. None of these issues, on their own, were extraordinary. Premature babies often have complications. But as the months passed, the Greenberg family began to notice something else. Brooke was not growing. At 6 months, she looked the same as she had at 3 months. At 1 year, she still looked like a small infant. Other babies were sitting up, crawling, beginning to babble. Brooke was not. She remained quiet, immobile, and tiny. Pediatricians ordered tests. Hormones were measured. Bones were X-rayed. The conventional culprits were checked one by one. Growth hormone deficiency. Negative. Pituitary disorders. Negative. Russell-Silver syndrome. Negative. Cornelia de Lange. Negative. Every test came back inconclusive or normal. Whatever was happening to Brooke did not match any condition described in the medical literature. And then it got stranger. Most growth disorders affect the body uniformly. A child with dwarfism, for example, may be small but proportional. Brooke was not proportional. Her body was aging in fragments. Her bone age, as measured by X-ray, hovered somewhere around that of a 10-year-old. Her teeth were the baby teeth of an 8-year-old, and they never fell out. Her brain, by contrast, was still developing at the rate of an infant. She never gained the language skills of a 1-year-old. She never developed object permanence in any reliable way. She could recognize her family. She could laugh. She had clear preferences and emotions, but the cognitive milestones that normally arrive between ages 2 and 5 never came. Her organs were the same puzzle. Some appeared the appropriate age for a child her chronological age. Others looked like the organs of a much younger child. It was as though her body had been assembled from parts of different ages, and none of them had decided who was in charge. Through all of this, Brooke endured medical catastrophes that would have ended most lives. At age 4, she suffered a stroke. At age 5, doctors discovered a tumor in her brain. The tumor disappeared without treatment. Her physicians could not explain it. She had ulcers that perforated her digestive tract. She slipped into deep comas for weeks at a time and then woke up. Unchanged. Her family began to call her medical history a series of impossibilities. She kept surviving things she was not supposed to survive. And she kept failing to grow when she was supposed to grow. We are halfway through this case, and the most disturbing finding is still ahead. By the time Brooke was a teenager, her family had been turned away by countless specialists. No one knew what to do with her. Some doctors suspected a chromosomal abnormality that had simply never been described. Others suggested an undiagnosed metabolic disorder. The standard genetic tests of the time all came back without conclusive findings. This included chromosomal microarrays and the limited gene panels available in the 1990s and early 2000s. The Greenbergs were left raising a child who seemed less and less like a developing human, and more like a fixed point in time. Photographs of Brooke at ages 1, 5, 10, and 16 are indistinguishable. The same round face. The same small body. The same uncomprehending gaze of a baby. Her chronological birthdays accumulated. Her biology did not seem to notice. The case began to attract attention from researchers who specialized not in pediatrics but in aging. If a body could fail to age in childhood, what did that say about the mechanisms that drive aging in the first place? Could a clue to slowing or even stopping the aging process be hiding inside Brooke Greenberg? The most prominent researcher to take up her case was Dr. Richard Walker, a developmental biologist at the University of South Florida College of Medicine. Walker had a working theory. He believed that human aging is driven, at least in part, by the orderly progression of developmental stages. We are programmed to develop from infant to child to adolescent to adult. Aging itself, in Walker's view, is a continuation of that developmental program. If the developmental clock could be paused, aging would pause as well. Brooke Greenberg seemed to be a living example of what that would look like. Walker named her condition Syndrome X. He proposed that Brooke had a defect in the master regulator of developmental progression, whatever that turned out to be. With permission from the Greenberg family, Walker and his collaborators began an exhaustive genetic investigation. They sequenced Brooke's full genome. They sequenced her parents and her sisters as comparison. They looked for mutations in every gene known to influence growth, development, hormone signaling, and aging. The results, when they came back, were astonishing in their lack of resolution. Brooke's genome contained no obvious causative mutation. There was no single broken gene. No clear deletion. No chromosomal rearrangement that explained her condition. Whatever was holding her in eternal infancy, it was not the kind of simple genetic error that geneticists had been trained to look for. Walker began to suspect that the answer lay not in a single gene but in the regulation of many genes. The way DNA is read, switched on, and switched off across the lifespan. The field of epigenetics, the study of these regulatory marks on top of the genome, was still in its early years. The tools to fully investigate Brooke's epigenome did not yet exist when she was alive. The Greenbergs, for their part, treated Brooke as exactly what she was. Their daughter. They cared for her at home with the help of nurses. Her sisters loved her. She had a wheelchair, special clothing made for her tiny frame, and a daily routine built around feeding, bathing, and play. She had never gone to school. She had never grown into the next phase of childhood. She lived in a kind of permanent first year. Brooke Greenberg died on October 24, 2013. She was 20 years old. The official cause of death was complications from her underlying condition, which had never been formally named beyond Walker's working term, Syndrome X. With the family's consent, samples of her tissue were preserved for ongoing research. Scientists are still studying her cells today. Other children with similar conditions have since been identified. A handful of cases around the world appear to share at least some features of Brooke's syndrome. Researchers have begun comparing their genomes, looking for the common thread that may explain why a small number of human beings simply stop progressing through life. What is now clear is that human aging is not a simple, linear process. Different tissues age at different rates. Different organs follow different clocks. In a typical human, those clocks are synchronized, more or less, by master regulators that we do not yet fully understand. In Brooke, those regulators appear to have malfunctioned in a precise and selective way. Her case is now cited in aging research papers around the world. It is, in a strange way, one of the most important medical mysteries of the 21st century. Because if we can ever understand why Brooke's body did not age, we may begin to understand why the rest of ours do. Next case. A man whose skin changed color over 15 years. The transformation was permanent. The cause was self-inflicted. And by the time he realized what he had done to his body, there was no going back. His doctors knew exactly what had happened. He had refused to listen.